In progress.
On a 34, 39, ouais, ça augmente encore un peu. Et il est 31, peut-être attends une minute de plus.
Écho, là.
Il y a un écho. Ah.
Okay, good morning, everyone. Thanks a lot for coming. We have an echo, no?
The voice from out is great.
Ah, ok, c'est normal du coup.
It's going to be annoying.
Okay, we start again. Sorry for that. So thanks a lot for coming. So we're going to start. So as usual, those of you who attend online, you can report any technical issue in the chat, and you can ask your question, please, in the Q&A tab, and we will allow you to interact during the discussion session with the room. And with that, thanks, Luis, for introducing this.
Okay, so hello everyone. We're quite happy today and honored to have Catherine Peichel as a speaker. So Katie did her PhD in Princeton working on mice, but then moved on to working on sticklebacks during her postdoc in Stanford. And since then, she kept working on sticklebacks, doing all kinds of interesting things on the genetics of adaptation, mixing with ecological work, ecological context, as you'll hear today. Since 2016, she's a professor at the University of Bern, where she's now head of the Evolution and Ecology department. So, well, I think I met Katie 2014 during a meeting, and I thought this was really interesting when what she said, and then I realized, oh, actually, I had lots of her papers in my literature. So probably it's the case for lots of you. You've came across her work, and even people doing. I know people here doing macroevolution on fish, etc. Lots of people have something interesting to hear, I think, today. So with this, I leave you the floor. So does she need that?
Okay. Perfect. So thank you, Louis. I hope I have something interesting to tell you today. Thanks for very much for coming in again for the invitation to be here. So maybe my title is a little bit provocative today, and we can talk at the end whether I've told you anything about evolutionary predictability, but I'd like to at least discuss that. So the big question is really whether evolution is repeatable. And it's maybe a little bit of a cliche, but this question was sort of raised by Stephen Jay Gould in his book Wonderful Life, where he sort of set out this now famous thought experiment. If we went back to the beginning of life on Earth and sort of hit the rewind button and sort of went back to the beginning of life on Earth and sort of so-called replayed the tape of life, would life as we know it on Earth have evolved? Would we have evolved? And for Gould, I would say the answer was mostly no, because there's too many sort of contingent or chance events that happened that sort of biased the direction of evolution. But we know that evolution does to some extent repeat itself, because we see the repeated evolution, for example, of placental and marsupial mammals, so very similar forms, independently evolving to adapt to very similar habitats, or these famous examples of cichlid fish that have adapted to similar habitats in two independent Great Lakes in Africa, where again, you see sort of very similar forms of fish independently evolving. So evolution is, to some extent, repeatable. Which leads to before I go on, I just want to define a couple of terms when we talk about repeated evolution that can have some, people argue about what we mean by convergent and parallel, but I just want to define what I mean for my talk today. For my system. So basically we mean repeated evolution is the independent evolution of similar forms in similar environments. And convergent, I would say, is when things evolve from different ancestral phenotypes. So this phylogeny of marine mammals is a nice example here. So we have 3 groups of marine mammals that have evolved from independent ancestors. Whereas parallel would be evolving from a similar common ancestral phenotype. So that would be the case of dolphins and whales, for example. And the system that I study, sticklebacks, we use the term parallel evolution. And so I'll be using that today. So just to keep in mind the terminology. The other important point I wanted to make here is that this independent evolution of similar phenotypes in response to similar environments is often interpreted as strong evidence for a role of natural selection. But I would say that this has not always been tested completely. And so in the first part of the talk, I'm going to actually do a test of the role of natural selection in repeated evolution, and then we'll move on to questions about the genetic basis of that. So I think this question of sort of, is evolution repeatable? Yes, it is to some extent, but then the question is really why? Why do we see these repeated patterns? And here's where I'm going to make the argument that's been made by others, that to some extent, if we can understand why evolution repeats itself or why it doesn't, that might allow us to predict how populations will respond in the future. And that's something, again, we can discuss in the questions and later today. But if we could predict these evolutionary responses, of course, that's relevant for sort of applied things like for medicine, agriculture, and conservation. Okay. So to address this question of why evolution is repeatable, I'm going to take you to these beautiful lake stream systems. So we have lakes and their adjoining streams on Vancouver Island in British Columbia. And in these lakes and streams live my favorite model system, which is the 3-spine stickleback. And so in these small, slow moving streams, we tend to find sticklebacks that look like this, that have sort of deeper bodies. And they are adapted to feeding on small macro invertebrates that live in these streams. And in the adjacent lakes, which tend to be deeper, we tend to have sticklebacks that look like this, that are much more streamlined, and they tend to be adapted to feeding on zooplankton that's living in the water column. Okay. And so we see these repeated patterns in lakes and streams across the Northern Hemisphere, where the stream sticklebacks tend to be more deeper bodied, lake sticklebacks tend to be more narrow bodied, but it's not always perfectly repeatable or perfectly parallel. And so why? Why do we see that sticklebacks in some lakes look more similar to sticklebacks in other lakes versus sometimes they're actually a little bit divergent? And to address this question, we thought we could take advantage of the sort of variation in the extent of parallelism to sort of understand why things are repeatable sometimes and not other times. Okay, and so this work is a collaboration between Dan Bolnick, Andrew Hendry, and Rowan Barrett, and that's actually sort of an older collaboration that I'll talk about. And here we are pretending to work, which is usually what the PIs do, is we sort of hang out and pretend to work, and then the other people actually do the work. So in this case, I really want to highlight Yoel Stewart, who actually led the work that I'm going to talk about. And 10 years ago, actually, we mounted this big field experiment or a field expedition to study 16 of these lake stream pairs on Vancouver Island. And we focused on Vancouver Island because we wanted to sort of minimize other variables. So this is all in sort of a relatively small geographic area. And so we could sort of look at patterns of parallelism within this specific area. We collected from each of these 32 populations or 16 pairs, we collected extensive environmental data. So data on flow, temperature, zooplankton abundance, benthic invertebrate abundance, et cetera. We collected morphological data, and we collected genomic data, in this case, RAD-seq data from each of these populations. Okay. So just to point out, this is the map of Vancouver Island, and these were sort of each of these 16 systems was an independent watershed. And in fact, when you do the phylogeny, you find for the most part that lakes and streams are each other's closest relative to each other. So these are really, we think, independent evolutionary events, this divergence into lake versus stream. There were a few exceptions, but for the most part, they were independent. So then we looked at the traits. So now what I'm showing you is sort of trait by trait. So for each of the 60 or so traits we looked at, what was the effect of habitat? So this would be sort of the parallel component, lake versus stream. And then we looked at the effect of the habitat by the specific pair. So this is sort of interaction between lake versus stream and the specific watershed. And so traits that we see up here with a high habitat effect are sort of evolving more in a parallel or repeated manner, and traits evolving somewhere over here have some effect of habitat, but there's also some idiosyncrasy, some differences among populations. And so there's actually relatively few that that show up as highly repeatable or parallel. And this is just showing sort of what I mean, and maybe in a more intuitive way. So for example, for gill raker number, which the fish use to process prey, so we know that this should differ between benthic and limnetic or lake versus stream sort of habitats. We see that in general, lake fish, so each of these dots is an independent lake population, and then the line connects it to its stream population. And so, for example, here for 14 out of the 16 pairs, we see that lake fish have more gill rakers than stream fish. And then there's two populations where there's not a lot of difference. So this is a highly parallel trait that was in that top left corner. And then body depth is the other trait that really shows a parallel pattern where, for the most part, again, stream fish have deeper bodies than lake fish. But there are, again, a couple exceptions. Here's a trait for which there's sort of a lot of idiosyncrasy. So sometimes the lake fish are bigger, sometimes the stream fish are bigger. And then here's a trait that just doesn't show a lot of variation among the populations. Okay, so traits vary. So we get a lot of variation. Then we sort of wanted to look, just put everything together and you get this sort of hairball. So this is just principal components, and again, the lakes are colored in blue, and their streams are colored in green, and you can see the lines connecting them. Okay, and it's just a mess, right? You can see sometimes lake fish are over here, sometimes they're over here. There may be some parallelism. Can we make any sense of this sort of giant mess? And to do this, we actually took an explicitly sort of geometric approach to parallelism. And so we measured two things between any two pairs of populations. So again, the lake population would be here in multivariate space, and its cognizant lake or stream population would be here in green. And then you can make a vector between these two populations. And then between any two pairs of populations, you can measure the angle between these vectors. And you can measure the difference in length of the vectors. So the angle sort of tells you about the extent to which these populations are evolving literally in parallel or at orthogonal angles. And the distance and length between those vectors tells you the extent of divergence between any two pairs of populations. So you can sort of, for each pair, you can take each pairwise comparison. So Roberts Lake Stream versus Boot, and then Roberts versus all of the other 15 populations. You can do this for the ecological data, you can do this for the phenotypic data, and you can do it for the genetic data. And then you can ask whether there's any sort of correlations between divergence, angle of divergence between genetic and ecological, et cetera. And so I'm just gonna give you the punchline, because this has been published for a while now, and say that we can explain the patterns we see. So even though we have this sort of mixture or this mess, if you just look at the PCA, that angle of morphological divergence is correlated with the angle of ecological divergence. So the degree to which we see these parallel or repeated patterns is to some extent driven by natural selection. So the more ecologically similar two lakes are, the more similar the phenotypes are. Maybe, I mean, this is obvious and this is sort of consistent with a prediction, but to my knowledge, it's sort of one of the first examples of sort of really testing it. The angle of morphological divergence had no correlation with genetic divergence, but that overall magnitude, that difference in the length of those vectors, is correlated with genetic divergence. And again, maybe not surprisingly, this suggests that the extent to which a lake and a stream can diverge is constrained by the extent of gene flow. So if populations are more phenotypically divergent, they're also more genetically divergent. And there is this opportunity for gene flow in the system. Okay. So, you know, in this case, why is evolution repeatable? I would say that repeated phenotypic evolution in the system does indeed reflect to some extent the action of natural selection, but there's some sort of constraints in terms of the extent of gene flow that also play a role in the patterns we see. All right, so now I'm going to turn from sort of the more ecological work to something that's probably more closely related to the work that I've done for many years, which is understanding evolution at the genetic level. And we know that to some extent, we find these patterns of repeated evolution at the genetic level. And these sort of very classic example of seeing differences in pigmentation between, say, lizards and mice is often due to mutations in this melanocortin-1 gene. And so, sorry, that's not showing up super well up there, but basically, so there's these, and a lot of times when people find that the same phenotype or similar phenotypes evolve and they're underlined by the same genes, those sort of are the things that get published. And maybe sometimes when you find it's not the same. And so many years ago, we sort of set out to ask a couple questions, which is how often is it really that the same genetic changes underlie repeated phenotypic evolution? And then why, more recently, we've been delving into the question of why. Why might some genetic changes be used more frequently when the same phenotypes evolve? Okay, so I'm going to start first with just the how often question. And to do this now, this is now over a decade old. Dolph Schluter and Matt Arnegaard and Gina Conti and I just did a meta-analysis of sort of combing the literature to ask, were these MC1R stories sort of the exception or the rule? And to do that, we just did a sort of a literature search, and we found, and this was really focused on studies where people looked at repeated evolution of phenotypes and then looked at the underlying genetic basis. So we weren't including sort of population genomic studies. And there were genetic mapping studies and then candidate gene studies. We actually captured a large diversity of taxa and a large diversity of traits. And again, I won't go into the details, but we calculated this, what we call the probability of gene reuse. And so the simple way to think about that is, let's say there's a trait like pigmentation. There are two genes that have been discovered to control variation in pigmentation. If the two comparisons that those two genes are used in both comparisons, probability of gene use is 1. If neither is used, probability of gene reuse is 0. If one out of the two is shared, it's 0.5. So on the left, I'm showing you the genetic mapping studies. And in the open circles are what we call cases of parallel evolution. So evolution from a similar ancestor. And in closed circles are the cases of convergent, what we call convergent evolution, evolution from different ancestors. This is somewhat correlated, not surprisingly, with the age of the divergence. And so overall, we see about this proportional similarity is average about 0.32. If you look at more closely related or parallel systems, it's much higher. And if you look at kind of these convergent cases, the proportion of probability of gene reuse is lower. These numbers are somewhat higher if you look at candidate gene studies, and this is probably because of this publication bias. If you know MC1R is involved in one system and you look in another system and you see the same, you might publish it more than not. But overall, I would say that this is relatively high proportionate. I think a surprisingly high proportion of gene use. Now, there's some caveats, of course, which I've sort of mentioned, this publication bias. It's a relatively small number of traits. Genetic studies or sort of genetic mapping studies tend to be biased against identifying loci with relatively small effects. So we're sort of tending to look at things that have larger effects on phenotypes. And it's a meta-analysis, so things were done in different times in different ways. So actually, I think there's probably been a lot of work in the past decade since we published this. And so Maida Bohutinska is a really fantastic postdoc in my group, and she and I are actually going back and trying to update this more with the intention of sort of getting to also the why questions of why might particular genes be reused more often than others. So stay tuned for that. Okay, so that was sort of the meta-analysis, and now I want to tell you what we've done in our stickleback system to also address this question of how often are the same genetic changes used. And we've taken two approaches, so both QTL mapping and population genomics, and I'll walk you through both of those. So here we focused again on freshwater stickleback species pairs. And so I've talked to you about these lake stream systems where we've studied these 16 pairs. And also we study these benthic limnetic species pairs, which I'll give you a little bit more information on the next slide. But basically these have evolved at least 5 independent times, also in the area around Vancouver Island. Sorry, it's not showing up so well. But basically, again, we're looking at a very similar region of the world here. So these benthic limnetic pairs are found within lakes. And so within the same lake, you have one member of the pair that's adapted again to living in nearshore habitats and eating macroinvertebrates. Again, they tend to be sort of deeper bodied and have a lot of differences in their craniofacial structures for eating these types of food. And then the limnetics, again, are sort of more streamlined and living in the open water of these steam lakes and feeding on zooplankton. And again, this is very similar to the divergence I told you about between lake stream sticklebacks, where the stream fish are adapted to eating macroinvertebrates in these sort of more vegetated habitats, and the lake fish are living out in the open water eating zooplankton. Excuse me. So the first approach was what we do a lot of in my group, which is QTL mapping. So we basically take individuals that have from populations that have very different phenotypes. We cross them in the lab, and then we do mapping crosses where we basically look at phenotypes and genotypes and look for regions of the genome where we see associations between genotype and phenotype. And then in some cases go all the way to identifying the genes that underlie the traits of interest. Today, I'm just going to focus on this part, which is just asking where in the genome do we find these associations. Okay, so for the benthic limnetic pairs, we had one cross from Paxton Lake and one cross from Priest Lake, and we had 58 morphological traits. The first question was just how much phenotypic parallelism is there? So in this case, most of the traits actually have diverged in parallel in these two pairs. So meaning they're sort of diverging in a similar way in the same direction between benthics and limnetics. There's a few traits that diverge, so in one lake, but not the other. And then a few traits that sort of diverge in the opposite direction in the two lakes. So for example, the head is larger in benthics in one lake and larger in limnetics in the other lake. And then we asked, okay, how often do we see that the same genetic loci underlie these similar changes? And what we found is actually about half of the QTL that we identified were shared. So they were identified in both Priest and Paxton as underlying this variation in the same trait. We found another close to 40% that just was a QTL in one population, but not the other. And then a few that were sort of QTL in the opposite direction. So the benthic allele makes a larger head in Paxton, and the benthic allele makes a larger head in Priest. I should also say that this is all for the QTL for traits that have actually evolved in a repeated way, so only for the parallel traits. So this is a relatively high proportion of QTL sharing. And this is sort of similar to our meta-analyses where we would be in this parallel evolution range. And we find about 40% of the time the same genes are reused when we use this proportion of gene reuse. So again, consistent, I think, relatively high amounts of genetic sharing. Now the story actually changes when we look in these lake stream pairs. So here we actually looked at 4 independent lake stream pairs, and we actually did several crosses within each lake stream pair. I won't go into the details, that people can ask me about it. We had about 60 traits. So here, again, we use this sort of framework to look at how parallel was divergence in these traits. And again, we see relatively few traits that have this sort of highly parallel divergence. So similar to what I showed you before, body depth and Gillraker numbers. And most of the traits sort of showed more of these sort of interactions between habitat and the specific watershed. So again, there's relatively low, relative to the benthic limnetic system, there's much lower levels of phenotypic parallelism. And indeed, we find that relatively few of the QTL are the same. Now, this is a little bit complicated. So for example, if we take the 4 different watersheds here, so for example, in Boot, we were looking at plate number is the trait. So in this case, within Boot, within the 2 different cross families, we found that chromosome 7 has a QTL. So this is a sharing of basically 1 out of 1 or 1. Then if we look between Boot and Pi, we find that in Pi, we have one QTL on chromosome 2 and one on chromosome 7. So the between watershed sharing here is 0.5. And then if we look over at, for example, Misty, we see that nothing is shared between Misty and Boot. So you can sort of do all of these different pairwise, crosswise interactions. And for plate number, among watersheds across all 4, we find that the proportion of shared QTL is 0.4, which is relatively high. If we look across all of the traits, we find that this proportion of sharing is around 0.15. So relatively low amounts of sharing of QTL relative to the benthic-limnetic system. And again, if we sort of put this in comparison to the meta-analyses, we see that it's sort of lower than in other systems. But I would say that if we just do a sort of permutation test to ask, is this more than you would expect by chance? It is. So we would expect sort of less sharing by chance than we actually see. So it's low, but still sort of significant. Okay, what am I doing? Okay, so from our QTL mapping system, I found what was interesting here is that the answer we get depends upon the system that we're looking at. Now I'm going to quickly walk you through some population genomics we've done on the same systems. And this was done by Diana Renison when she was a postdoc in my group, and she now has her own group in California. So for the lake stream data, we had, again, these 16 lake stream pairs, sequencing across a bunch of individuals. And then for the benthic-limnetics, we looked at 3 of these benthic-limnetic pairs and sequencing data for a similar number of individuals. And then Diana just calculated FST in windows across the genome and basically asked how often was it the same for different windows. And so I'm just going to show you the overall patterns. So for the benthic-limnetic pairs, I hope what you can appreciate is you could almost superimpose these patterns on each other. So if we just look across the 21 stickleback chromosomes and then FST across these 3 benthic-limnetic systems are very, very similar. The same regions of the genome are basically showing high FST between the pairs. But if you look at the lake stream pairs, it's not so similar. So the overall levels of FST, just absolute, are different. And then where we see these sort of divergence is also not very similar across these pairs. So again, how often do the same genetic changes underlie repeated evolution? Sometimes, but not always. And in this case, it differs among our stickleback systems. So now I will briefly turn to the question of why. Why might it be that some genetic changes are used more frequently when similar phenotypes evolve? And there's a few possibilities here, not exhaustive. But it might be that for particular phenotypes, there's only one gene or one mutation that could actually produce a phenotype that's favored by selection. In general, for most of the phenotypes we study, we don't think that that's really the case, but it could be for some very specific phenotypes. It could also be that many genes or mutations could produce the phenotype, but that some sort of are less pleiotropic, and therefore meaning that they affect many, many traits and then maybe have fewer constraints. These are not mutually exclusive, by the way. It could be that they're in regions of the genome which have higher mutation rates. So for example, in stickleback, repeated evolution of the loss of the pelvic spines, which are found on the body of the fish, is actually due to repeated mutation at the same gene, this gene called PIDX1, which David Kingsley's lab at Stanford has actually shown is in a fragile region of the genome that's actually highly mutable. So this is a possibility. Or it could be that things are just in regions of low recombination, and so which could produce a biological reason that things sort of that need to be linked together are, or that you just detect them because they're in regions of low recombination, which is a caveat, for example, of QTL mapping. Also, of course, of FST. So Diana and I tried to look at this in these population genomic data sets that I mentioned before. And so Diana basically characterized for her benthic limnetic data. So parallel windows would be those windows that were differentiated in 2 or more of the 3 pairs. And then the non-parallel windows would still be regions where we saw differentiation, but only between 1 of the pairs. And then similarly for the lake stream data, like I said, there was overall less parallelism, but we could still find sort of these parallel windows. So again, we used windows that were differentiated in 2 or more pairs. And then we have the non-parallel windows that were differentiated in just 1 of the 16 systems. And then we basically asked if these differed in different genomic features. So for pleiotropy, we use 2 proxies, which I fully admit are proxies, because to really measure pleiotropy, you'd have to do the knockouts, you'd have to put the fish in all the different environments and see, like, is this gene, actually want to make the actual mutation, not just knock the gene out. So we basically use two sort of proxies for pleiotropy. One is the number of QTL that have been previously mapped to that location of the genome. And we have a database of over 1,000 QTL that have been mapped for a variety of traits in stickleback. So that was one. And then the other was gene connectivity. So using RNA-seq data, you can sort of look at gene co-expression, and if a gene is co-expressed with more other genes, this has sort of been argued to be a proxy of pleiotropy. They both have their pluses and minuses, but we use both to try and sort of see if we saw similar patterns. We then asked also if these windows differed in gene number or recombination rate or mutation rate. So here are the results. So what we found, so I'm showing on the left, so these are the stream lake data, the benthic limnetic data, and then the distribution of number of QTL in non-parallel and parallel windows. And in both of these cases, the parallel windows had twice as many QTL mapped to those regions as the non-parallel windows, which was actually in contrast to what we had predicted, that parallel sort of regions that were involved in repeated adaptation would be less pleiotropic. And we actually found that on average, they were more pleiotropic. But I will note, especially here in the benthic limnetic data, that the most pleiotropic loci here are in non-parallel windows. We saw a very similar pattern for connectivity as our measure of pleiotropy. So again, parallel windows tended to have a higher level of connectivity in their genes than non-parallel windows, although the most sort of connected windows are here in the non-parallel. So do these windows differ? I won't show you all of the data, just to say overall, yeah, parallel windows have more QTL and higher connectivity, but are the most, but non-parallel windows have the genes with the highest level of our proxies of pleiotropy. There's no difference in gene number between parallel and non-parallel windows. In recombination rate, parallel windows do have a lower recombination rate, but this is only true in the benthic limnetic pairs where we know there's lots of gene flow. And if we just consider the outlier windows, we don't really see a difference. So we don't think recombination rate is driving these patterns. And there's also no difference in sort of mutation rate if we take into account recombination rate. So from these data, we sort of conclude that pleiotropy doesn't always seem to be a constraint on the repeated use of similar loci. And rather, we would say that probably intermediate, or especially if it's synergistic in terms of its fitness effects, levels of pleiotropy might actually be adaptive. So consistent with the fact that these fish are adapting to this, they're adapting to predators, to prey, to potential abiotic conditions. And so if there is a locus, it sort of allows sticklebacks to evolve to adapt to this multivariate environment that could actually be adaptive. And this might be particularly important in systems like our sticklebacks, where we know that a lot of adaptation is due to selection on standing variation in the population, and that these alleles are old. And so if you have these sort of older alleles that have been repeatedly tested by selection, these sort of synergistic pleiotropic alleles might be the ones that are sort of maintained and repeatedly used. Okay, so just super quickly, we've also been thinking as well about the role of gene flow in contributing to patterns of when similar phenotypes evolve. Why might some genetic changes be used more frequently? And so MIDA has been doing a meta-analysis of population genomic data. So there's actually Heliconia's papers, a paper from Patrick, and one of MIDA's papers on Arabidopsis that sort of suggested that in populations that are more closely related, where there's an opportunity for gene flow, you actually get more repeated gene use. And so MIDA looked at this in a bigger meta-analysis, and I won't go into the details here, but basically to show that with increasing divergence time, you see a decrease in gene reuse. And these studies here in green are where you see this sort of negative relationship between gene reuse and divergence time. And this is pretty strong across a wide range of divergence times. But then when you start to get into these older divergence times, the pattern goes away, and we're starting to think about sort of mechanisms for that. So I'm happy again to discuss this with people. So probably there is some role of pleiotropy and gene flow, and probably some interactions between them on why and whether we do see these patterns of gene reuse. But this is really something I would love to continue to think about and work on in the future. I don't think we have the full answer there. Okay. So I just want to come back in the last couple minutes to this question of why do we see these really different patterns in the benthic, limnetic, and lake stream pairs, where we see sort of high repeatability at the genetic and genomic level in benthic limnetics and relatively low in the lake stream pairs. This is sort of highlighted here. If we just look at these FST correlations, so if you just take, for example, Paxton benthic limnetic FST and ask for a given window, does it have low or high FST? And then we plot this relative to, say, Priest, and we say if a window has high FST in one population pair, does it have high FST in the other? And the overall correlation for this is 0.5 across the benthic limnetic pairs, consistent with sort of those graphs look very similar that I showed you earlier. It's about 0.15 for the lake stream pairs, which is pretty similar actually to the QTL data. And then maybe I was surprised by this. So the actual, when populations are evolving to benthic habitats in the streams versus benthic habitats in lakes, there's really almost no sharing between these two different systems. So, why is this? Is it that they have different sources of standing genetic variation? I don't think so. These are all populations from Vancouver Island. The marine ancestor, which is sort of the source of standing genetic variation, should be the same for these populations. Is it something about the evolutionary history, how they were colonized? We don't know. The extent of gene flow does differ. So benthics and limnetics probably have more gene flow than lake stream. Or it could be something about the strength of selection, of parallel selection. So there's this nice paper by Ken Thompson that suggests that as selection is more parallel, you sort of see an increase in the extent to which you see genetic parallelism. This is maybe supported by the fact that we see a lot more parallel phenotypic evolution in the benthic limnetic pairs and less in the lake stream pairs. But all of this is sort of still just thinking about ideas. And the problem for most of us as evolutionary biologists is that we're historians. We're sort of looking back in time and trying to say what happened, and we don't know exactly what the founding populations were, and we don't know how those ecosystems are changing over time. We're just looking at what they look like now, but they might have gone through changes. And so, what if we could actually do Gould's thought experiment that I raised at the beginning of the talk? And of course, many of you in this room are doing exactly these experiments. So the classic example being Rich Lenski's sort of evolution in bacteria. So you set up repeated colonies and sort of passage them over time and ask, do the same things happen at the phenotypic and genetic level when you run these experimental evolution experiments in the lab? But, of course, these are super important, but in these cases, we're always sort of, manipulating a particular, maybe a single variable, maybe a couple of variables. We're working in the lab. And so what if we could actually do these experiments in natural ecosystems and really try and get at to what extent do we see repeated evolution in natural ecosystems when we sort of know the starting conditions? This is exactly what we're doing actually in lakes in Alaska. So in 2019, we had the opportunity to put sticklebacks into 8 empty lakes in Alaska. So Alaska Department of Fish and Wildlife, there were invasive pike in these lakes. And so they used rotenone to kill off all of the fish in the lakes, but then they wanted, as part of a restoration process, wanted to put sticklebacks into these lakes. And so they came to Mike Bell, who studied sticklebacks in Alaska for 30 years, and said, hey, would you like to decide which sticklebacks we put in these lakes? And he said, well, actually, I'm about to retire, so you should talk to some of the younger people, not so young, in the field. And so a bunch of us got together and decided on an experimental design. So 4 of these empty lakes were sort of smaller and more benthic in their habitat, and 4 of these lakes were larger and supported sort of a more limnetic habitat. So then we went out to the area and identified 4 source lakes that had benthic lake fish and 4 source lakes that had limnetic lake fish. We used 4 source lakes to sort of maximize genetic variation. And so we made a pool of benthic fish and a pool of limnetic fish. And then 2 of those benthic pools, we seeded benthics into 2 benthic lakes, benthics into 2 limnetic lakes, and vice versa. Limnetics went into benthic lakes and limnetics went into limnetic lakes. And so this was done in 2019. Remarkable. So we were able to actually collect from these lakes in 2020 and 2021, despite COVID. And I can say that the introductions were super successful. There are thousands and thousands and thousands of sticklebacks in these lakes. And then I got a grant from the Swiss National Science Foundation. So the EU kicked us out of the ERC sort of grants, but Switzerland, being Switzerland, made their own ERC thing, and that really allowed me to get money to sequence the genomes of every single one of these fish that went into these lakes. So we have whole genome, we will, hopefully by the end of this year, have whole genome sequences and phenotypes. We took photos of all of those fish from all the founding fish. And then the plan is just follow sort of evolutionary trajectories of both genotypes and phenotypes in these lakes, and we'll be able to do at least 8 generations. And of course, we also have a whole team of people working on the ecology of these lakes, so we can also see how these things are changing over time. This is just one of Andrew Hendry's drone photos showing these actual lakes. And the idea is to ask, how repeatable are the trajectories that we see over these 8 generations? And then, this is the more challenge. Can we predict? So knowing the genotypes of every fish that went in, who ended up successfully mating, how ecological factors are sort of changing over time, can we actually predict what we what we find, having sort of full information, as full of information as we can have. So with that, I will just stop there, and just again, I try to mention everyone who I'm working with in this, but yeah, I'd be happy to take any questions if there are any.
All right, thank you, Katie. So questions in the room? Yes, Johan.
Hi, thank you for the talk. I was wondering because at the end of the talk, you mentioned a lot of time the gene flow. And so I was wondering if there is more or less gene flow between different systems. Where do you put the line between parallel evolution and the evolution of one metapopulation that is just one population evolving and with more or less gene flow?
Yeah, so good. That's a super good question. So the first question is like, there is a lot of variation in gene flow, and that's something we haven't done, is to go back and actually see how that variation in gene flow affects these sort of genomic patterns. That's something I'd like to do. So are the patterns just because sticklebacks are just one big meta population? I would have said yes to that, except for then what's happening in these lake stream pairs? It's the same standing variation, right? The same source populations here. Why in the lake stream pairs don't we see this massive sort of repeatability at the phenotypic or the genetic level? So until I got those data, I would have sort of agreed with you, like, yeah, of course, okay, sticklebacks, repeated evolution, of course, it's just selection on standing variation. Maybe once in a while, it's just repeated mutation at the same locus. But these lake stream guys are making me sort of, I just don't know.
But in the lake stream, is there not less gene flow compared to limnetic, benthic
There still is
populations?
some gene flow between lakes and streams, and it varies across the lake stream pairs. We think they were founded in the same way by the sort of marine ancestors. So yeah, if people have ideas, I would love to hear them.
Okay.
Yeah.
Thank you.
Mhm.
Thank you, Katie. Is this working? Yes. I have a question about the point you made about parallel selection. You found that certain traits, like certain trait category, are more parallel than others,
Yeah.
like feeding, for example. So do you think that those are under stronger selection than other category of traits? And if yes, do you know, or has this been looked at, that is there a relationship between the strength of selection and the likelihood of parallelism?
So, okay, so for like in the lake stream pairs, it's definitely body depth and Giller-Acre number, sort of really clearly under strong selection, strong parallel selection. I didn't show the plot here, but I plotted in the lake stream data the degree of parallelism at the genetic level and the degree of phenotypic parallelism. No correlation. No correlation. So more strongly parallel traits are not more genetically parallel, at least within the lake stream system. Yeah. No. But then I think maybe the overall degree of parallel selection is higher in the benthic limnetic system. So that could be one explanation for why it's potentially just more genetic parallelism as well. You don't detect that within the lake stream data. Mm-hmm.
Hello again. I've got a little methodological issue with how you identify parallel. Well, we don't know which phenotype is really under selection, and you probably know this example with Drosophila, where with UA study where Drosophila were introduced in North America, and there you could see that they re-evolved larger wings, I think,
Mm-hmm.
and as it turned out, it was instead of having well, you can evolve larger wings either by having larger cells or having more cells.
Mm-hmm.
But the key point is that you got a larger, let's say, wing overall. But if you don't know that, and you have a bunch of traits, and you don't have the idea or to combine on some traits which is actually the target of parallelism. And then you do your
Yeah.
PCI, and then you will miss that and think that it's all over the place, because sometimes it's the number of cells, sometimes the size of those cells, and so you miss the signal about parallel evolution that would be there if you were to identify the trait. So how do you sort that kind of?
Yeah, I mean, it's an absolutely the issue, right? Is like we don't know what the actual target of selection is. I mean, for things like Gill-Raker number, we're quite confident that that has something to do with how the fish capture their prey, and we know sort of these form function relationships. But there are many ways. I mean, but that's, I think the question is there one way to make more Gill-Rakers, or are there many ways? And then when you see that it's the same locus, you can say, okay, they do it the same way. And then when it's a different locus, they're clearly making things in different ways. But it's always defining your phenotype that's a target of selection, I think is a hard question. I mean, it's just, and that's what we also try to do it across many traits to do that. But I think that's the interesting, I mean, that is then you start to get into like, why is it in some cases it is the same, in some cases it's different. What's the cellular phenotype, for example? What route does it take to get to this phenotype? So I hope that sort of answered.
I had a related question about pleiotropy. I was wondering, was there a correlation between your different proxies for pleiotropy that you use, connectivity and number of QTLs? Did you look at that, or? So
just in those windows, were they correlated? That's a good question, because I don't think we did. I mean, I would say probably yes. I mean, the phenotypic data, the QTL data, I can't remember what tissue the gene expression data was for. So sort of a mix of different tissues, which may or may not be correlated. But I wish we could look at that easily. No.
Okay, so to close it first.
About gene flow and the genetic architecture of traits and the divergences, those predictions that if you've got a lot of gene flow, only a locus with big effects will actually stay polymorphic and contribute to divergence, while if you have little gene flow, you can actually have much more idiosyncratic genetic architecture, because there might be many more small effect genes that can be recruited in less predictable way in different places. So do you see any evidence of a pattern like this?
I mean, I think that the benthic-limnetic lake stream comparison does a little bit, because I think there's more gene flow in the benthic-limnetic system than in the lake stream systems. But again, it varies across the lake streams. But then overall FST is really high in those benthic-limnetic systems, but it's really restricted to these particular regions of the genome, which is why I have a hard time sort of really counting gene flow, if you know what I mean. Because if you just look at overall FST, you would say it's way lower in the benthic-limnetic systems, but it's concentrated. Or sorry, it's way higher in the benthic-limnetic systems, but it's really concentrated. And then in the lake stream system, it's distributed. But the overall levels are variable. So I think if we can look within the lake stream system, we can try to address that a little bit better.
Thanks, Katie. I had a question about environmental variability, because the underlying idea or basis behind parallelism is that there's parallel selection, right? And in that first study, you actually showed that there was this spectrum of parallelism, but it correlated with the amount of ecological divergence. So to what extent, I mean, basically there's variation among lakes and there's variation among streams. So could another way of thinking about this be actually there's a lot more parallelism than maybe your talk lets on? It's just not between lakes and streams. It's in correlation with the quantitative variability among lakes and streams. And that's what that first Nature EU paper basically implies, right?
That's what it says, is that there's a correlation between, it's not just lake stream divergence, but just the overall patterns of phenotypic variation that you see are correlated with ecological variation. And so if a stream is more lake-like, the fish look more lake-like. Yes.
So then the second question, is there more parallelism than your talk maybe lets on? In that context?
Maybe we'd have to look at that, actually. That's an interesting question. If you plotted, for example, some ecological variable, if we use that in our linear models, would we explain more of the sort of repeated variation that we see than just this coarse lake stream sort of habitat that we did?
Yeah, and you could try the phenotypic level at the
A genetic level. Yeah, no, that would be, yeah, I think we could do that. Or someone smarter than me could do that.
Other questions online, perhaps? People are listening or in the room? No? Okay, well, with that, I just thank you again for the talk.
Thanks, everybody.